Oh, do you know that just like money is needed for transactions in the real world, cells require a currency for transactions, which we call high energy compounds. I'm Dr. Juliet, and welcome again to Jules Summit Biochemistry Hall, your go-to place for mastering biochemistry one concept at a time. Today, we are looking at the cellular currencies, which is molecules, compounds that help cells to perform their processes by supplying them with the needed energy for sustaining life. What are high energy compounds, which also called cellular currencies? They are special molecules that act as energy traps. What does that mean? Is either they contain very unstable energy-rich chemical bonds, or they carry energy-rich electrons from where they are produced to where they are needed. These high energy compounds are critical to the sustenance of life because without them, cellular processes cannot take place. They will come to a halt, and that will mean the end of life itself. Basically, we have three major classes of high energy compounds, the currencies of the cell. Number one will be phosphorylated compounds, high energy phosphorylated compounds like ATP, PEP, and PC. What are these? These special molecules contain energy-rich phosphate bonds. ATP is the most popular among them since it's almost present or partakes in chemical reactions that require energy. So ATP is a pyrophosphate compound. We have enol phosphates like phosphoenol pyruvate, which we see as one of the glycolytic intermediates. And then we have the phosphagen phosphocreatine. These phosphorylated compounds, like I said before, they contain energy-rich phosphate bonds that are very unstable, and when they are lysed or split, they release significant amount of energy which cells can use for their processes. The next major class will be thioesters. Why are they called thioesters? They are esters, but in the place of oxygen atom, which we find in esters, these ones contain sulfur atom. An example or examples would be acetyl-CoA, S-linosylmethionine, and succinyl-CoA. These two, as we know, play major functions in fatty acid metabolism and the tricarboxylic acid cycle. And then lastly, we have reducing agents like the nicotine adenine dinucleotide and the flavine adenine dinucleotide. These ones do not contain high energy phosphate bonds like the first two, but rather they contain high energy electrons whose transfer from one molecule to another releases large amount of energy that drives chemical processes. Phosphorylated compounds and thioesters are popular as high energy compounds because of the high instability of their bonds. These bonds breaking up releases large amount of energy, which scientifically we say they have large negative Gibbs-free energy change during their lysis, which means their breakdown is very favorable energetically and produces large amount of energy. But the third class, like I said, do not contain high energy bonds, but rather they serve as delivery couriers, carrying high energy electrons to places where they are needed for the production of energy, particularly for the conversion of ADP to ATP, the ready or most popular energy currency of the cell. Why are these high energy compounds so important? They store energy. Remember, initially we said they are energy traps. They hold onto energy in bonds, which are very unstable and easy to break, releasing the potential energy they've been holding, which cells can trap and use for their processes. So they store energy and they transfer energy. If we relate this to the glycolytic pathway, we see ATP, an example of energy compound, coming in at the first step, converting glucose to glucose phosphate. So you see that they supply energy where it is needed for chemical reactions. And also they transfer, like we see in the last step of the same glycolytic pathway where phosphoenol pyruvate and enol phosphate releases energy for the production of ATP and thereby being converted to pyruvate. So they store energy, they also transfer energy. An example again is in the TCA cycle where we see the energy of the intermediates, the energy of the glycolytic intermediates being released into reducing agents like NAD, H and FADH2. These will now carry this energy to the electron transport chain where they donate it to the proteins there for the conversion or for the buildup of the protein gradient that later drives the phosphorylation of ADP to ATP, our most popular energy currency. And then lastly, what the other function is they drive processes directly. They are involved in active transport. They are involved in muscle contraction, and they're involved in cellular respiration, and even the biosynthesis of macromolecules.